Hi, this is Hillary. My hair pulling concept is the basic structure of the bacteria cell wall. I'll be going over some differences between gram-positive and gram-negative cell walls, and then separately but also closely related and important, I will be talking about the plasma membrane in either type of cell. And just so there's no confusion, the plasma membrane is actually not part of the cell wall. So that's why in my drawings I've highlighted the cell wall as yellow and the plasma membrane is represented in orange. Okay, so bacteria are most commonly either gram-positive or gram-negative. Gram-positive cells are definitely different than gram-negative cells, and it's distinguishing characteristics in cell walls that will indicate whether bacteria is gram-positive or gram-negative. Important features in a gram-positive cell wall, you'll notice this really thick layer of peptidoglycan. So that big yellow chunk there in my drawing would represent that structure. So that peptidoglycan layer helps the bacteria maintain its shape and it provides structure. There would also be an inner wall zone between the thick peptidoglycan and the single membrane. So the wall zone would be somewhere right about in here. Two main components of the plasma membrane. It has a lipid bilayer, so you can see kind of the two layers, top and bottom. And different types of proteins are found in the plasma membrane. Lipoteichoic acids would be an example of a protein found in the plasma membrane. These anchor through the peptidoglycan layer onto lipids that are on the lipid bilayer. So those would kind of go through the peptidoglycan. So it would be lipid to lipid here. There's also another type of teichoic acid not found in the plasma membrane, but found in the cell wall. This is called wall teichoic. So this would hold the peptidoglycan together. I don't know if you can read my handwriting. So the wall teichoic acid would be kind of inside of the peptidoglycan itself, but not going into the plasma membrane. Okay, so those are some qualities of the gram-positive cell wall. Let's take a look at the gram-negative cell wall. So first off, I'll just start by talking about the plasma membrane in a gram-negative cell. So a gram-negative cell also has a plasma membrane, and this is still composed also of a lipid bilayer that contains proteins. It also has a layer of peptidoglycan, but you can see in the drawing here, this layer is very much thinner, so it's much thinner than the thick peptidoglycan in the gram-positive. So this is a key difference between gram-positive and gram-negative cells. Another difference is that the peptidoglycan layer is anchored to an outer membrane. So it has a second lipid bilayer. I'll just zoom in a little bit. Now, the outer lipid bilayer contains proteins called porins. So the porins would be found in this outer lipid bilayer. Porins allow for passive diffusion of things from the outside of the cell into the space between the two membranes. or what's called the periplasmic spaces. And those are drawn a little bit right here, periplasm. Another important feature of gram-negative cell walls is what's on the very outside of the outer membrane. LPS, or lipopolysaccharide, is a component only found on the outside of a gram-negative cell wall. It contains several structures like O-polysaccharide and lipid A, which is also known as endotoxin. Endotoxin can induce fever as well as an immune response in the body, or you could consider the fever as the immune response. Okay, so just in general, some key terminology for the cell wall and also the features that we've discussed so far between gram-positive and gram-negative. Peptidoglycan. So again, this is a protective layer for bacteria. And it's composed of amino acids and glucose. So proteins and sugars, two molecules in peptidoglycan, acetylglucosamine, otherwise known as NAG, and also acetylmuramic acid, otherwise known as NAM. So to combine the two molecules together, we would need the enzyme called transpeptidase. Trans means across, so basically these two molecules are cross-linked, and this forms a thick layer of peptidoglycan. Well, it actually forms the peptidoglycan in both gram-positive and gram-negative, but since it's cross-linked, it's strong. Again, lipoteichoic acid is only found in gram-positive cells, and this provides cell wall rigidity. It also, similar to what we talked about with lipid A or endotoxin, it has antigenic properties, and it's able to stimulate an immune response or a fever. It would basically recruit leukocytes in the body as the body detects an invader. The periplasm that we talked about with mostly we discussed in the gram-negative cell. Just to clarify, both gram-positive and gram-negative have periplasm, but it's going to definitely be more obvious in the gram-negative cell wall. And then we also talked about porins. These are found in the outer membrane. They're like little channels that allow things to come into the membrane and other things not to come into the membrane. So for example, bacteria love glucose, and they would definitely allow that to come in. They would not want, for example, antibiotics to come in. So there are some bacteria that have gotten resistant and have learned how to block out antibiotics, which come in through the porin and kill the bacteria. This advancement keeps the bacteria from being susceptible to being killed by certain drugs. An example of an antibiotic is penicillin. This prevents peptidoglycan from forming, and it makes the cell wall useless and vulnerable to rupturing or lysis in gram-positive cells. It's a little bit different scenario with penicillin in a gram-negative cell, and the reason for this is that the gram-negative cell has a second membrane, so it's not as affected by penicillin. So that wraps up a brief overview of differences between the gram-negative and gram-positive cell. mostly the cell wall, and then we also looked at the plasma membrane as it exists in either cell. Thank you so much for listening.